In the modern age, we tend to take for granted all of the work that goes into bringing food to our table. We can easily drive to the store and pick out whatever we want, vegetables, grains, fish, or even clams, but take a step back and think, how did those clams end up in that store? 89% of commercially sold bivalves come from aquaculture operations, making it one of the fastest growing and economically important sectors in food production. Florida's shellfish aquaculture industry developed in the 1980s in response to a still unresolved natural hard clam stock collapse. Shellfish aquaculture heavily relies on deploying shellfish into the marine environment in order to grow to a marketable size. Growing shellfish in their natural habitat is extremely cost effective. However, it also leaves them vulnerable to consumption by common mollusk predators. In the process of harvesting aquacultured clams, clammers have reported crushed shells and torn gear, speculating that white-spotted eagle rays are to blame for this economic loss. But how can we begin to understand if these rays are interacting with hard clam enhancements versus other mollusk predators in the area? Quite simply, we can sample white-spotted eagle ray stomach contents to see exactly what they're feeding on. Because natural hard clam populations are limited, we know that if we observe hard clams in the stomach contents of these rays, that there's a high likelihood that the rays are foraging within areas designated for shellfish aquaculture. White-spotted eagle rays are highly mobile mollusk predators common to Florida coastal waters. While previous work suggests that white-spotted eagle ray diet is variable throughout their western Atlantic range, a comprehensive diet analysis has not yet been conducted in protected Florida coastal waters. In order to effectively describe their diet, I'm employing two techniques: DNA barcoding to directly identify stomach contents, and stable isotope analysis to provide insight to longer duration assimilated dietary patterns. Using non-lethal methods like pulse gastric lavage, we are able to analyze stomach contents using traditional visual identification and molecular barcoding. Preliminary information from 15 white-spotted eagle rays collected in Sarasota, Florida suggests individual diet variation. However, we have positively identified hard clams in a portion of those stomach contents. We're currently working to analyze an additional 37 stomach samples in order to more thoroughly assess the frequency of these potential interactions. When used in conjunction with traditional diet analyses, stable isotope analysis can provide insight to the food web ecology of white-spotted eagle rays, highlighting which prey items such as aquacultured hard clams are most important to their diet. Thus far, I've collected plasma, red blood cells, and muscle biopsy samples from 68 rays and muscle samples from 304 potential prey items. By measuring nitrogen and carbon isotopic levels across all samples, I can determine the proportion of prey items that were consumed and assimilated into the ray tissues. While sampling and analyses are ongoing, these data will provide important information on the trophic role of the state protected species. Not only will the findings from my study fill important knowledge gaps for white-spotted eagle rays, but more importantly, it will assess these animal interactions with growing aquaculture industry and hard clam restoration initiatives along Florida's coast. Thank you.